Good afternoon. As you know, I'm Francis Wright. I know most of you. It's an honor to be here as a recipient of the Dean's Prize for Summer Research. And today I'll be talking to you about my summer research in the Marsden Lab, spying on SPY1 for patients with an immunodeficiency disease called A-gamma-globulinemia. So, to start with some background, collaborators with the Marsden Lab brought us 5 interesting cases of patients stricken with an immunodeficiency disease called A-gamma-globulinemia, or A-gamma for short. These 5 patients have recurrent, life-threatening lung and sinus infections throughout their lives, and they have low numbers of antibodies in their blood, which are an important thing that B cells release to protect us from pathogens. The interesting finding was that on exome sequencing, all of the genes that normally cause A-gamma were wild type. The only mutated genes were in SPY1, which is an interesting gene, and if this is causing their A-gamma-globulinemia, this could be the first ever SPY1-related immunodeficiency ever described in humans. Bringing your attention to the bottom of the screen, there's 4 black boxes and 1 black circle. These represent the 5 patients with A-gamma. Importantly, they're all unrelated. They all come from different families, and they're heterozygous for SPY1 mutations. All of these patients have sporadic mutations, meaning that they have not been passed down throughout their families. And in this diagram of the protein that SPY1 encodes for, you can see that their mutations occur in different areas of the protein. These are 2 important domains for protein function, and we'll get into that more later in the presentation. So more about SPY1, what we do know is that it encodes for a protein called PU1. In humans, we know PU1 acts as a tumor suppressor. And we know from mouse models that it's very important for B cell and macrophage development. We're not sure how it functions for the human immune system, and so that's a question we want to answer for these 5 patient cases of immunodeficiency. The Marsden Lab is well poised to tackle this question because of our interest in studying the genetics of immune cell function, and our specialty using a tool that you may have heard of called CRISPR. It's widely used for editing and studying the human genome. And so, the hypothesis of my summer research is that mutations introduced with CRISPR into human stem cells will decrease their ability to fully develop into mature and functional B cells. This would possibly recapitulate the immunodeficiency that we're seeing in these 5 cases. So the first step of my SPY mission was to genetically engineer human stem cells with mutations in the SPY gene. You can see at the top here, this is a Sanger sequence, which represents the DNA at the SPY1 locus. Each colored peak is a different nucleic acid in the DNA, and the dotted line you see there is where CRISPR cuts so that we can edit the genome. At the bottom of the screen is the sequence of the genetically engineered cells. You can see mixed peaks after that dotted line because we now have a heterogeneous population of genetically engineered cells with random insertions and deletions that cause frame shifts and delete the function of the SPY gene. And we also still have a population of homozygous wild-type cells that are not edited or touched by CRISPR at all. So that was the first step. The second step was to take this heterogeneous population of genetically engineered stem cells and wild-type cells and differentiate them in the lab into mature B cells. So we did that, and we used something called IgM, which is a surface protein only on the surface of mature B cells, and we looked for that as a sign of full maturity. We used a technique called fluorescence-activated cell sorting to purify a population of IgM-expressing cells. And the next step was to take that pure population of IgM-positive cells and sequence their genomes to look and see if any of the genetically engineered cells that we made with SPY1 mutations fully matured into IgM-positive B cells. So for reference again, here is the wild-type sequence at the SPY1 locus. And taking this population of IgM-positive cells, this was the main finding of my summer research project, and it's very important to me because what we found is absolutely none of the genetically engineered cells fully developed into IgM-positive cells. From that heterogeneous population, only the cells that were homozygous for wild-type SPY1 could fully mature. And I think this is an important finding because it lends evidence to the fact that maybe these patients with this condition where they constantly have lung and sinus infections have this disease because of their mutations in the SPY1 gene. So I think the next steps of this project are to look at each of these patient mutations in more detail. And it's also, I believe, important to develop CRISPR-based gene therapies that could potentially correct SPY1 mutations back to wild type. It could return their B cells to functioning, and these would parallel the FDA-approved genetic therapies that are available now for severe combined immune deficiency. So these have a lot of potential if we develop these to really improve the lives of these 5 patients and any patient with a SPY1-related immunodeficiency. I really want to thank Alex Martin, David Wynn, and Amy Chen because they taught me an enormous amount this summer. They were my teachers, my mentors, and my friends, and I could not have done this without them. Thank you.